Greetings to everyone. Today we would like to present the topic on chemical engineering for sustainable food futures. We are from University Putra, Malaysia, Group TVC01, the Power Puff Girls. Our group members are Shim Shu-si, Ang Choyen, and Anna Q. Ting. Firstly, we will start with the future potential of lab-grown food. Our team believes that lab-grown food will leverage the food industry, and we will explain it for three aspects, which are sustainability, biodiversity conservation, and global health. For the sustainability, lab-grown food has the potential to be more sustainable than traditional agriculture because it requires less land, water, and resources, and it has the potential to reduce the environmental impact associated with livestock farming, such as the deforestation and greenhouse gas emissions. Moreover, it could contribute to climate change mitigation as well. As I said just now, by reducing the demand for large area of land for agriculture, that growth could potentially help conserve natural habitat and biodiversity, as it decreases the need for deforestation and habitat destruction associated with traditional farming practice. Since the nutrient in the lab-grown food could be controlled and it could provide a more controlled and sanitary environment for food production. So this could help reduce the spread of disease associated with conventional livestock farming, such as the zoonotic disease. Other than that, it has the potential to reduce foodborne illness risk and also boost patient recovery by providing an adequate and sustainable protein.
Consumer acceptance is heavily influenced by the taste and texture of food. Lab-grown products must replicate the sensory experience of traditional meat to gain widespread acceptance. Continuous improvement in the taste and texture of the lab-grown food is essential for overcoming initial skepticism by chemical engineers. Next is the unnaturalness and draw comparisons to artificial or manufactured food in the public become one of the challenges facing by the chemical engineers. Furthermore, the challenge in the misunderstanding of genetic modification by the public. Some may associate genetic modification with altering the fundamental DNA of an organism. However, the current processes for culture meat production typically involve cell culture rather than genetic modification. Next, the safety of vegetables cultivated in laboratories can be jeopardized by microbial contaminants, including bacteria, fungi, and also growth-promoting agents, fertilizer, or pesticide. To guarantee the safety of the finished product, chemical engineers need strict adherence to aseptic procedure, monitoring and control of nutrient solution, caution uses of chemicals, and comprehensive testing for both microbiological and chemical pollutants are necessary.
The following part is about the current advancement in the field of lab-grown food, which is 3D bioprinting. It is the integration of microfluidic technology and a bioprinting platform to create a composite tissue using 3D model computer data through a layer-by-layer self-assembly technology. However, 3D bioprinting using bioink instead of traditional inks and materials. Bioink consists of hydrogels, which are a combination of biomaterials, cells, and other biomolecules of interest used to construct the products in a 3D printing process. To print a 3D food product, the bioink is loaded into a 3D bioprinter, and the printer will follow the digital models to deposit the bioink layer by layer. This layering process allows for precise control over the arrangement of cells and other components. In 3D bioprinting, the primary methods are as follows. The first one is inkjet printing, which is able to produce ink droplets with controllable size and low volume. The bioink will deposit in specific locations with high precision and without contact. Employing this technique, it is possible to use a variety of biomaterials and the incorporation of living cells. The following method is extrusion-based bioprinting. The bioink in this method is also deposited with high precision by obtaining customized 3D structures such as customizable forms, pores, porosity, and cell distribution can all be controlled precisely with good structural integrity due to the continuous deposition of the elements. The entire process is carried out under the control of a computer. Last but not least, wet photopolymerization is also one of the methods of 3D bioprinting. It is an emerging technology in the biofabrication of scaffolds applied in tissue engineering. This method using light-activated polymerization to actively curing photopolymer liquid resin. The molecule of liquid photopolymers quickly bond together and cure into a solid form when exposed to a specific wavelength of light. This process known as photopolymerization is all for current advancement. As for the solution, we are also deciding on 3D bioprinting technology as the solution of the taste and texture problem. In 3D bioprinting, there is a chance for texture enhancement by engineering tissue-like structure, controlling cell density and distribution, and also it can optimize the composition of hydrogels. There are also quality control and contaminant prevention, which are sterilization techniques and real-time monitoring are present in 3D bioprinting. Together, these steps allow for better food security.